everybody, it's Ms. Hunt, and today is Monday, the 30th, and I hope everyone is doing great. We're down to 3 weeks left. Yay, I'm so excited. Okay, I hope you had a great break and you're ready to rock and roll for the next 3 weeks. Okay, just a reminder for my online kids, according to our school, we are not doing online school second semester. So if you are a virtual kid, you have to come back in order to stay in honors and AP classes. Otherwise, you will be switched over to Edgenuity from Whitfield County Online Academy, and they will handle you for biology, but you won't get the honors credit. So if you want to come back and get the honors credit, I hope to see you in January. Okay, we're going to click into classwork. We're in module 13 this week. Everything's a little slow today. Well, here we go. Okay, let's go into our lesson plans first and take a look at this week. So this is what we've got today. We're doing notes for 9.3 to finish up mitosis. You should have these notes already from last week, but I've attached them again. And we're finishing our study guide, which would be great if you could turn that in today electronically, or if you are an absent kid and still have the packet, I'll take it when you get back. So turn in that study guide today, finishing it up for homework if you need to. Okay, tomorrow we have it's a dry lab, but it's a worksheet, but I'll take it as a lab credit. And you're going to go through an online platform, but it's on our classroom. The loss of cell cycle control in cancer cells. It's a digital assignment. Okay. Wednesday, we're doing new notes, and I've got them here in the classroom, and a study guide to go with it. But we're just doing 10.1. That's it. We're just doing meiosis. Thursday, we're going to draw meiosis. So that should be a lot of fun to draw that all out. And then Friday, you guys have a POGIL where you have to read and try to work your way through meiosis. All right, next week, we're doing reproduction. And then we have our exam for mitosis and meiosis for the cell cycle. I put it over 2 days because usually my exams are pretty big and they have a written component on it. So you'll have over 2 days to get that done. This also gives us oh my goodness, Ms. Hunt's tests are kind of hard. So if you started on Thursday and you need to study a little bit more on Thursday night, make sure you really know what you're doing, and then finish it up on Friday. That's okay. You can go back and revisit it. The following week is our midterm. So we will take our midterm on Tuesday of the following week. We'll finish and do reteaching and any kind of test corrections to try to gain some points back. Those kids who are failing might have some work over Christmas break to do on USA Test Prep. If you don't pass the class, then you have to take biology all over again. So hopefully you will do well and bring our scores up and we can have everybody passing. But I still have some kids who are failing. On the Friday before we leave for Christmas break is a half day, and I won't do any schoolwork on that Friday. So that'll be our free day, which will be really nice, a half day to be able to watch some movies. Maybe, I don't know what we can do. If we can try to get kids in doing something with us, that would be nice. And then, of course, that would be the last online day. So everybody will be coming back in January. All right, and then you'd be off for Christmas for 2 weeks, or Christmas break. Okay, let me go back in here. Today, we finished up Chapter 9 notes. So let me go through Chapter 9. And let me first do, my normal reviewing. We're studying in mitosis. We're studying the cell cycle. So we studied cell growth, and we said, cells had to grow because they couldn't get too big. Because if they got too big, then they would be inefficient, slow, and things couldn't move through them very easily. We studied mitosis and cytokinesis. And now we're in that last section of cell cycle of section 3. Okay, let's put this up here. Okay. All right, now, let me move my face here. First of all, you should recognize this picture up here, right there. Do you recognize what that's representing? Okay, hopefully you recognize that that's representing the cell cycle. And do you remember there's 3 parts to this cell cycle? Interphase, mitosis, and cytokinesis. Very good. Do you remember the 3 parts of interphase? G1, S, and G2. Very good. All right, remember in interphase is the longest period of the cell. In G1, my cell grows and it does its job. In the S phase, that's where we synthesize DNA. Make sure you remember that is where DNA gets made. So everything that we learn from the DNA unit, for the DNA helicase unzipping, the RNA primase priming, the DNA polymerase building, and building the 2 strands, a leading and a lagging, that was semi-conservative replication. All that happens here in the S phase. All right, G2, the cell prepares for mitosis, and then we have mitosis. Remember we said PMAT, prophase, metaphase, anaphase, telophase? At the end of here, we have cytokinesis. Remember we pinched our cell and said cytokinesis, okay? Hopefully you already remember all of that. If you didn't, go back through and read the book, go back through and watch my videos again to make sure you know what you're talking about. Okay, in our new notes today, we're going through a protein. So let's first remember, let me get paper because I love to draw. Okay. Okay, hang on. You remember how we have enzymes that are specific, and then we have the substrate that binds to the enzyme, and they bind in the active site called the enzyme substrate complex. Okay, hopefully you remember that. So our enzyme this time is kinase. Remember, kinase ends with an ACE. Okay, so kinase, ACEs are usually enzymes. Okay, and our substrate is cyclin. Okay, so it really looks like this. So what happens when we go through our cell cycle? We have little checkpoints to make sure that the cell is doing what it's supposed to do. I made an analogy like when I get in my car. Have you ever gotten in the car before and you forgot to buckle up, but the car went bing, 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 and your mom or dad said, buckle your seatbelt's not on. And you remember to reach over and buckle your belt. Okay, so that's like a safety checkpoint where you stop and you fix your problem. Same thing like when we're driving. Maybe we see a miles per hour sign, a speed limit sign. And we say, well, I need to check my speed. Am I matching the speed limit? Am I going over? Am I going fast enough? Make sure I'm doing the right thing. So we kind of have these checkpoints built in. And then we get in our car and we put it in park and we go to turn it off or turn our key. Has your car ever binged because you left your lights on, or binged because you left your keys in the car, or done something? So again, there's another checkpoint. So it's very similar with our cell as we go through this picture, which is the cell cycle. All right, we're going to get these little stoplights, which are cell cycle checkpoints, okay? And what happens at that checkpoint, okay? At the checkpoint, kinase, which is our enzyme, comes here. And if everything goes right, cyclin will bind to kinase and it'll allow our cell to go to the next level. So after G1 is done, where our cell's growing, we have a checkpoint. If everything is good, then kinase will bind to cyclin and you will go to the next phase. The next phase is the S phase, where we synthesize our DNA. So if everything checks out in this phase, we synthesize our DNA correctly, then we go to the next phase, to G2. And this is all the same thing. The cyclin binds to the kinase and we pass on to the next, okay? G2, everything's growing, ready for mitosis, for cell nuclear division. Everything's good, check, go to the next one. Even after mitosis, after PMAT, prophase, metaphase, anaphase, telophase, we check, did everything go right? Okay. Now, the cool thing is, if it's not right, your body can fix it. Your cell will fix it. The cell will go back and fix that DNA. The cell will go back and make sure all the organelles grew the way they were supposed to. So everything checks throughout this whole thing and is able to fix it. And then finally, we get cytokinesis. And we pinch apart and we have two cells, okay? So what do you need to know? I would like for you to know the word cyclin-dependent kinase. You will see it as CDK sometimes. It's nice if you can think on it at a higher level, that this kinase depends on cyclin. And if you see it as an enzyme substrate complex, that the kinase depends on the enzyme, the kinase depends on the substrate cyclin to bind in order to make the complex to allow it to go to the next thing. That's great. But if you just know these checkpoints are regulated by cyclin-dependent kinase, then we're good. The quality control checkpoints, the cell cycle has built-in checkpoints that monitor the cell and can stop if something's going wrong. Spindle fibers have also been identified in mitosis as checkpoints. Something else my kids had a struggle with today. Make sure you remember mitosis is nuclear division. This is when the nucleus divides. This whole thing is the cell cycle for a new cell, for two new cells, two new daughter cells. But this mitosis is just when the nucleus divides in the cell cycle, okay? All right, so if you make it through all of that, so let me go back right here. If my cell grows and it makes it through the checkpoint, but it didn't have something correct, but it still made it through, and my DNA was synthesized, and maybe there was a problem on the DNA and it made it through, and I go through growth 2 and I make it through, and I go through mitosis and I make it through, somehow I make it through all these checkpoints, then sometimes we can get cancer. If something was allowed to pass through because of a mutation, an error in DNA, damage to the DNA, damaged cell, anything like that could become cancer. Now, you should know cancer is extremely rare. I know we all know somebody who has had a cancer, but cancer itself is still a rare occurrence. Cancer is uncontrolled cell growth. So if I don't control my cell growth, what that means is the G1 phase is cut very short. The cell doesn't do its job. So it goes G1S, G2M cytokinesis. And it doesn't spend a long time in this G1. Everything happens really quickly. So you can see here, let's say that these are lung cells. These are just normal cells of your lung. They're supposed to take in oxygen and help you breathe. And one of the cells in this area mutated, and it became two daughter cells that were bad. Because cancer cells don't do G1, it's not going to do the lung's job. It just reproduces over and over. So we get this lump. The problem is that lump is taking space of cells that are supposed to be doing a job. They're supposed to be helping you breathe. And instead, they're not. So the person might have a hard time breathing. The other part is when a lump is found, they sometimes will decide whether it's benign, which means it's not doing anything, it's just a lump, or if it's malignant, which means it's spreading. So here, these cancer cells have hit a vein or an artery, and these cells are being taken away through the body, where they could attach to other parts. They could attach to the heart, they could go to your stomach, to your bloodstream, it could go to your brain. So that's where it gets really bad. So right now, this is just a lump. All right, and it's uncontrolled cell growth. So the checkpoints don't work, nothing is stopping it. Those cells just keep growing. Cancer can kill an organism by crowding out the normal cells, resulting in the loss of tissue function. Causes of cancer. The changes that occur in regulations and the cell growth and division of cancer are due to mutations. Various environmental factors can affect the occurrence of cancer cells. So a word up here that we should have put up here is carcinogens. Carcinogens. Carcinogens are things that cause cancer, and you'll read about this in the book. There's lots of things that cause cancer. One thing that everybody knows that causes cancer is tobacco. We know smoking cigarettes and doing tobacco causes cancer. So cancer caused by tobacco is a mutation, and you have exposed your lungs to something that's bad, it's a carcinogen, and it can cause cancer. There's lots of things that can cause cancer, and you can Google it, known carcinogens, and find a good reputable source to look at different known carcinogens. The biggest thing is we just want to limit our exposure to carcinogens. So UV light can cause carcinogens, so too much sun exposure, getting a sunburn, for example. Getting too many x-rays can cause cancer. And of course, exposure to drugs can also cause cancer. All right, here's a graph showing the life of a normal cell. So our cells should be growing, they should be doing their job, and then they should be dying. But a cancer cells, they're not going to die, they're just going to reproduce and grow faster and faster. So this is an uncontrolled cell growth. All right, another backup is called apoptosis. So if your cell recognizes that it's not doing what it's supposed to do, it can cause itself to destroy itself, okay? It's a programmed death of a cell. Sometimes we'll see the word cell suicide or that the cell kills itself. I don't really like those words because they're just not very sensitive. So I tend not to use them, but you might read that. So I just want to make sure you're aware. I would say that it's a programmed death of the cell. Apoptosis makes you think it's going to pop, but it doesn't pop. It really just shrinks down and shrivels up, okay? And then it stops and it will kill itself so that it doesn't do anything bad. It doesn't go on to make any more. So it's another backup, okay? Something else that gets regulated by the cell cycle is when cells are supposed to become other cells. Like when are they supposed to reproduce to become something different, which is crazy. We've talked about the cells being identical to themselves, but you didn't start off as all put together as you are right now. One day, egg and sperm came together and made one cell. And then those cells reproduced quickly to make a bunch of cells that were called stem cells. Those stem cells are going to change and become muscle, blood, nerve, bone, all the different cells of your body. All right, but at one point, embryonic stem cells, after fertilization, the resulting mass divides and repeats until about 100 to 150 cells that have not become specialized. So these right here are your stem cells that have not become specialized. So in that cell cycle regulation, the cells reproduce same until something turns on to cause it to change. And the new cell that's made is a muscle cell and a blood cell and a nerve cell. So something different, okay? Now, there's a lot of research in using stem cells to heal you. For example, what happens if I had this in my lungs and they cut this out? It'd be nice to regrow lung cell. If I put a few hundred stem cells here and they touched my cells, they would become lung cells. They would grow new lung cells for me, which would be great. Okay. There's controversy though. We don't want to use these cells because these cells are stem cells from fertilization. So they are from an egg and sperm coming together, and if they were left alone, they would become a baby. Instead, we're going to use these cells, which are adult stem cells. So these are found in various parts of your own body. Of course, they were found in your umbilical cord when you were a baby. So if your parents happened to save that, then you could use your umbilical cord. I know when I gave birth, they had a paper that said, preserve your kid's umbilical cord. But it was like thousands of dollars. So I was like, no, sorry. Hopefully they find cures before then. But in that umbilical cord would have been stem cells. In your body, it's like in your hips are some stem cells. So if I had a hole in my heart or damage in my brain, or maybe my spinal cord had a mistake or a break in it, and I needed to regrow those cells, then I would use adult stem cells from my own body and then have them put there. So scientists have to dig around inside of you and find them. And they're less controversial because they are not an embryonic cell. So they don't have the potential to become a baby. These are just hanging out in your body. Okay. All right. Now from here, you are going to also do the cell cycle study guide. And I'll tell you, the section 3 was a little bit challenging. Just going through the notes, it was a little bit harder. Okay, so we're just doing this today. So going through the notes made it a little bit more difficult, but when I read the book, it was almost verbatim. I could go through here and fill this out by the book. So make sure you're reading chapter 9, section 3, and you'll be able to do this no problem. Just use your textbook to help you out. Okay. All right, that's for Monday. I'm going to go ahead and do Tuesday so that you know what we're supposed to do for Tuesday. If I can get it to work. That'll load in just a second. Okay, Tuesday, you're doing the loss of cell cycle control cancer digital assignment, and you're going to turn that in online. So it like showed up for a second. Okay. That's this right here, mitosis cancer dry lab loss of cell cycle control. So you're going to read through this document, and it'll tell you like where to click on this. It's Khan Academy, which we've used before. And you're going to be able to write your responses here, reading responses and reading articles and filling this out. So as you go through, you should be filling it out and working on it. And that's what we're going to be doing Tuesday. Okay. On Wednesday, I will make a new video and go through new notes for you. And then I'll make another video on Thursday of me drawing. So this one should cover you for Monday and Tuesday. I hope you have a great day and a good week. Bye.